Good afternoon, ladies and gentlemen. Welcome to the announcement of the Brain Prize 2015. My name is Kim Krogsgaard. I'm the Managing Director of Grethe Lundbæk European Brain Research Foundation. With us today we have the Chairman of the Board, Professor Poul Krogsgaard Larsen, Chairman of the Selection Committee, Professor Sir Colin Blakemore, and Professor Maiken Nedergaard, University of Copenhagen and University of Rochester. Welcome. First of all, I'll give the word to Professor Poul Krogsgaard Larsen. Poul, please.
Thank you very much, Kim. Ladies and gentlemen, this is the day and the moment that we have been looking forward to, the announcement of the 2015 Brain Prize winners. Before I announce the winners, I would like to extend my deep felt gratitude to the distinguished members of our selection committee, chaired by Sir Colin Blakemore. Also this year, the committee has done a comprehensive and meticulous work. And this work is not only highly appreciated by the Foundation, it is the very precondition for the prize and its standing in the international neuroscience community. Without many good nominations and the collaboration with the International Neuroscience Committee and community, there would be no Brain Prize. Thus, we are indebted for the support shown by the many nominators and by our increasing network of international collaborators, that is, individuals, institutions, and organizations. The award of the 5th Brain Prize is a defining moment for the Foundation and the prize. The Brain Prize has come of age, and we hope that it will continue to grow and mature in terms of visibility and impact, as well as will our outreach activities nationally and abroad. Now to the announcement. The Brain Prize 2015 is awarded to Winfried Denk, Arthur Connert, Carol Svoboda, and David Tank. For the invention, refinement, and use of two-photon microscopy to provide detailed dynamic images of activity in individual nerve cells, dendrites, and synapses, thereby transforming the study of development, plasticity, and functional circuitry of the brain. They are not here, but congratulations to all of you. With these few introductory words, I wish to call upon Sir Colin Blakemore for stating the reasons for the 2015 Prize Award. Please, Colin.
Thank you, Pavel. It's a pleasure to be here to announce the winners of the Brain Prize this year, and I think that this award is another indication of the valuable contribution of Europe to the development of neuroscience globally, the importance of collaboration and interaction on an international scale in neuroscience, as you'll see from the backgrounds of the prize winners this year, and the way in which they have worked on both sides of the Atlantic to further this field. The prize this year has been given for the development, the refinement, and the use of a technique called two-photon microscopy. It has a complex technical background, but its importance and contributions are quite simple to explain. Their significance, I think, is best indicated by the fact that since the early 1990s, when the technique was first developed, more than 10,000 scientific publications using this method have been published. And it is now pretty much the standard technique for visualizing activity in individual nerve cells and groups of nerve cells, and has been applied very widely in neuroscience. Efforts to develop ever better methods of visualizing cells and what they're doing has really underpinned the development of neuroscience from the end of the 18th century, actually, most particularly and distinctively through the work of Ramon y Cajal, who won the Nobel Prize at the beginning of the 20th century for revolutionary descriptions of the structure and the organization of the nervous system. But until fairly recently, the last few decades, the methods for studying the detail of the nervous system were limited to dead tissue which had been treated to make it more transparent, easier to visualize with conventional light microscopic methods, but obviously limited by the fact that it was dead. The efforts to develop methods to visualize activity in living tissue have been really very important. The story begins with a young German, Winfried Denk, who first studied physics in Munich, but had before that, from an early age, been interested in working with his hands, making things. He helped his father in his workshop, where they lived near Munich, learning how to use a lathe, how to use simple instruments, designing mechanical things. He got fascinated in electrical engineering, largely through the contact with a professor of electrical engineering who was the father of a friend of his at school. And he started to design simple electrical circuitry and make electronics for himself. He studied physics in Munich, then went to Zurich to work for his master's degree in physics. And while he was a student, he took an undergraduate research position at the IBM laboratories near Zurich, at a stage when that particular laboratory was working on a variety of different microscopic methods. So he's interested in microscopy and in biological tissue as well, developed at the same time. He decided to do his PhD in the United States. He actually applied to Berkeley and was rejected by Berkeley, but perhaps fortunately for the history of neuroscience, he ended up in a very important department of physics at Cornell University, run by Watt Webb, an inventive and very important scientist who'd built a group working on various aspects of the physical limitations of microscopy. And he was also interested in the use of fluorescent methods for visualizing activity in tissue. In this background, Denk decided to study the activity of hair cells in the ear, the cells which respond to sound and which signal sound information to the brain. At that stage, the work in that area was developing in a very interesting way, looking at the mechanical properties of these hair cells which respond to vibration in the ear. But as a sideline, Denk developed a personal interest in the possibility of using a phenomenon called two-photon fluorescence in microscopy. And let me explain what two-photon fluorescence is, because it is the crucial background to the development of this technique. Ordinary fluorescence involves a fluorescent molecule, a substance, which itself generates light, it fluoresces, when light of a different wavelength hits it. It absorbs energy from the incident light and then emits its own light of a different wavelength, and that is the fluorescence phenomenon. Most fluorescent compounds respond to single absorptions of light, single photons of light, single photon fluorescence. But in 1932, Maria Goeppert-Mayer, who won the Nobel Prize in 1968, a theoretical physicist, first suggested that it might be possible to produce fluorescence in some molecules by the simultaneous or virtually simultaneous absorption of two photons of light if the light was intense enough, there was a probability that two photons would hit a single molecule at the same time. This was just a theoretical proposal, but later the realization of that phenomenon was observed empirically. It was possible to make fluorescent compounds that could be activated only by very, very intense light. And Denk had the idea that if high energy light could be focused at a point inside living tissue, that it might be able to activate fluorescence at that point alone and not in the surrounding areas of tissue through which the light had already passed. And the great advantage of that would be that the fluorescence would be emitted only from that one point and wouldn't be obscured by fluorescence that had been produced by other molecules on the way as the light passed through it. So that's one of the crucial features of two-photon microscopy. It achieves remarkable clarity of focusing of the image and clarity in the detail that's being visualized by the fluorescent method by virtue of relying on this two-photon absorption. Another crucial step was the use of infrared light. Infrared light isn't really light because we can't see it. It's just beyond the visible spectrum. It's beyond red. It's of a longer wavelength than red. So we can't see it with the naked eye, but it can be used to illuminate through a microscope. And the great advantage of infrared light as opposed to visible light is that it scatters less inside tissue. The longer the wavelength of light, the less the tendency for the light to be scattered, and therefore it produces less background illumination. And also, it can penetrate more deeply into tissue than short wavelength light can. So here are three advantages: deeper penetration, less scattering, and this two-photon emission limiting the image to a single plane. Another quite recent technique was the development of pulsing of laser light, meaning that very high energies of light could be deposited very, very briefly, and therefore the damage produced by extremely intense light could be limited. So the rapid pulsing, which had recently been developed too, was important. And finally, other microscopic methods had stimulated the development of scanning techniques in which the laser beam is scanned very rapidly in a raster pattern through the tissue, creating an image of a two-dimensional plane, and then eventually extending to 3 dimensions by scanning downwards through different planes. So the opportunity to develop this technique in the late 1980s, 1989 was the original invention, depended on the convergence of a wide variety of different scientific and technical developments. And Denk saw that if these things were combined and if the method worked, it could provide a technique for visualizing, using fluorescence, visualizing tissue, even quite deep under the surface of tissue. So with his colleagues, another graduate student, Jim Strickler and Watt Webb, he developed the first confocal microscope in 1989. The paper was published, I think, in 1990. He went back to Europe, but then decided to go to Bell Telephone Labs to continue this work a few years later. And the reason he chose Bell Telephone Labs was because one of the group directors there, David Tank, an American, had played a very large part in the development of various molecules, various substances, which fluoresced and which could be used in nerve cells specifically to visualize the activity of those cells. And the majority of these compounds depended on the fact that when cells are active, a small amount of calcium enters the cell, both from outside the cell and from storage vesicles inside the cell, and the calcium is then used to trigger other reactions in the cell. So if calcium can be visualized, if the appearance of calcium inside a nerve cell can be visualized, it's an indicator of activity in that cell. And that's been the principal way, actually, in which this technique has been used to study activity in the nervous system. So together, Tank and Denk worked in 1994 and first applied the technique to nerve cells. The first paper was published in 1994. The same year, they were joined by a young Czech researcher, Karel Svoboda, who had studied in the United States at Cornell, in the physics department of Cornell, and at Harvard studying biophysics. And he again was attracted to Tank's laboratory in Bell Labs as the kind of powerhouse of new methods for applying calcium indicating dyes to the study of the nervous system. And there was an extremely productive period through the middle of the 1990s where this trio worked together to establish the technique, applying it to a number of problems in neuroscience, on the structure of the dendritic processes of cells, even visualizing the individual tiny spines, less than 1,000th of a millimeter across, that stick out from the dendrites of cells, where the excitatory contacts, the synapses, end on cells. So the three of them together were able to work out the dynamics of the synaptic input to dendrites, the way in which activity spread through the dendrites, how it passed through bifurcation points in dendrites, how it led eventually to the integration of information in the cell body of the nerve cell before the generation of its own impulse going along its own axon or fiber. So each of them made very important individual contributions during that period, but they also worked together in a series of seminal publications. They've now gone their own ways, David Tank to Princeton, Karel Svoboda now at the Janelia Farm, the Howard Hughes Institute in Janelia Farm near Washington, D.C. And Winfried Denk back to Europe, now with an appointment at Munich as well as at Heidelberg in two Max Planck institutes. They've maintained their own interests and activities, developing further the application of this technique, applying it, for instance, to studying the way in which groups of nerve cells interact with each other as the circuit to the network of neurons is computing to calculate what action to carry out, what signals to send to the rest of the brain. And a crucial contribution to that particular technique was work done in Europe, separately from the other three, by Artur Konert. Konert was born in Romania, went to Munich to study medicine, worked in Göttingen in the Max Planck Institute, went to the United States for a postdoc for a short period of time, but is now in Munich and is a leading figure in the biophysics of neurons in Europe. His principal contribution in this area, though, was the development of a technique called bolus labeling, to distribute the labeling dye, the fluorescent dye, amongst a large group of neurons so that their activity can then be looked at simultaneously and in real time. The images produced by this method are usually dynamic. They show the changing pattern of activity. They're very beautiful. And it was Konert's contribution that really made that possible. Konert has gone on to make very important contributions himself, I mean, most particularly, I think, recently, looking at the way in which nerve cells in sensory parts of the cerebral cortex combine input and signals and information from the different synapses and dendrites before the final output of the cell is produced, which then represents a particular feature of the auditory input or the visual input received by the cortex. So these four people have contributed to the invention and then the development and particularly the exploitation of this technique to study the nervous system. Their work has ramifications through the whole of neuroscience, this now being an absolutely standard technique for studying structure and functional organization of the living brain. I should mention, and Maiken, speaking after me, will, I think, tell you more about this, the importance of not just the knowledge of physics and engineering methodology, as well as biophysics and biology, but also the understanding of the computational problems needed to interpret this information. And this is one of the big problems faced by neuroscience in the future, it's all very well to gather more and more information, to be able to see more and more nerve cells and how they're active, even the individual components of nerve cells. But the ultimate problem is, how do you interpret all of this? So the importance of computation, being able to compute, to process, and to understand the meaning of signals runs in parallel with the development of two-photon microscopy. I think it's no surprise that 3 of these, of our group of 4 prize winners are really very significant mathematicians and physicists themselves, and have been able to apply that knowledge as well to the interpretation of this technique. So we are delighted, we in the committee are delighted to have been able to make this award. And let me just point out finally, one of the reasons for our pleasure, the Brain Prize, the raison d'etre of the Brain Prize is to recognize the contribution of Europeans and Europe to the progress in neuroscience. But Europe doesn't work in isolation, and science doesn't work in isolation. I think this and some of the other prizes in previous years show very well how Europeans working with the rest of the world have helped to develop a field. And that's exemplified wonderfully by a Czech researcher, a German, a Romanian working in Germany, and an American working together in different combinations in different places, have pushed forward this really crucially important contribution to current neuroscience. Thank you.
Thank you, Colin. Thank you very much. And the last speaker this afternoon is Professor Maiken Nedergaard. Maiken, please.
Yes, I'm very happy to be here. And I wanted to say that neuroscience is probably the most fascinating of all sciences. Not only because it's very complex, but also because it's really the science about who we are and what little things go wrong when we have patients that suffer from schizophrenia or Parkinson. Before the invention of the two-photon microscope, we are actually limited to look at the brain with electrical recordings. You can probably best describe what a remarkable impact two-photon microscope have had on modern day neuroscience by looking at electrical recordings as a radio. And now we suddenly got a TV, and we can get direct images of what is going on in the brain. And that opens up a completely new way of looking at the brain that not only look at neural computation, but look at all the other 90% of the cell the brain is composed of that do not use electrical communication. And that is really a major breakthrough in my mind. So these investigators are all basically, as Collins described, they're basically all engineers. And they are able to do their remarkable studies for which they've got the Brain Prize because they're engineers. They could build their own setups. And they're actually not known. If you ask people in the field, you ask them, so what does Karl Svoboda do? They would say he's a neurobiologist who studies brain computation. They would not state, no, this guy, he was really one of the frontiers in two-photon imaging. So one thing I wanted to state was that I think that the Lundbeck Foundation have done a very bold and very important thing by nominating the topic of two-photon imaging as giving the Brain Prize. Because two-photon imaging has probably just surfaced at what it can do. Because to now, two-photon imaging have been used mostly for neural computation, because these were the people who were engineers who had a background in neural computation. With two-photon imaging, you don't only listen to the one who can speak, you can actually see all the ones that can speak. And these include microglia cells, it includes the blood vessel, it includes amyloid plaques. And with the conversion of not only fluorescence technique and laser technique, but with the conversion of transgenic mice model, where you can study Parkinson, you can study Alzheimer, and you can study in many different models schizophrenia. We might go in and find out what is going on. This technique would probably not be of major impact in patient research, but in the very, very basic research, this is an amazing contribution. From my own point of view, I actually started out visiting Karl Svoboda, I think in 1999, and he was incredibly generous, showing me that have convinced NIH to give me $500,000 to get a commercial two-photon scope. He was very generous to show me how you can build your own for $100,000. So and what happens then was that we were actually capable of building multiple setups that benefited a lot of people that study very different things from me, especially the blood vasculature. That the emergent concept is that the brain vasculature is probably one of the major contributors to dementia. So this was a key for me, basically interest in non-neuronal cells. The major contribution we have done to the field is that we ask the basic question, if the brain really is about no computation, why do we need to sleep? A computer, you really don't need to sleep, which is already telling you there's much more to the brain than you can imagine using multiple recordings. And here the two-photon microscope comes in, because what we did, and we used it in a very simple way, we basically just labeled this cerebrospinal fluid. And what we observed was that the cerebrospinal fluid almost acts like a dishwasher that enters the brain, but only when we sleep. And that might actually be the biological basis for sleep, because the brain cannot do the function other tissue can when we awake. The brain has to go into a mode where you can have the neural computation and you can integrate both input and you can actually have signaling between all the different parts of the brain within milliseconds that require incredible amount of sophistication, not only from the neurons, but also for all the supportive cells. I think Rodolfo Lina has probably stated that the best way, and that is when you have race drivers. You have one race driver, but he has a team of 20 people behind him, and he wouldn't be able to win the game if they are not doing the best job. And similarly, you can state of the brain cannot function at this sophisticated level as we are interacting now if you don't have the supportive cells and you don't take care of your housekeeping function during the function of sleep. And this is important because neurodegenerative diseases are intimately linked to disturbances in sleep, and they might actually progress the development of the disease. So this is why I feel very deeply about that. This was a very important decision to devote the Brain Prize of 2015 to a technique that have so much promise. The final thing I wanted to say is that it's also converging, and this is why the prize is so important right now, converting that you can actually now purchase these instruments for fairly reasonable price. And that means that experts in different diseases that are not engineered can very easy adapt the technique and combine it, for example, with electrical recording or blood flow analysis or even behavior. Just yesterday, there was a publication in Nature that suggested that mice, if they're touched by another mice, if they expect they're touched by another mouse, they actually respond before they're touched, and nothing else can replicate that. And you can go in with two-photon microscope now and figure out which are the cells that actually were acting before the touch occurred. And then I'll just congratulate the winners and for this. Yeah, so thank you.
Thank you very much, Maiken. I think that was it. Thank you very much for coming, and thank you to all of you out there who followed this event live at our website. And also from me, congratulations to the prize winners.